Hello. I'd like to start by thanking the organizers for the opportunity to present my work on microglia and aging. My name is Sara Mason. I'm a PhD student at Case Western Reserve University, and my main interest is understanding the process of neurodegeneration and how HIV infection in microglia leads to transcriptomic changes associated with aging. As we all know, there are several types of cells in the brain that work together to maintain homeostasis and optimal brain function throughout our lifespan. These include neurons, astrocytes and oligodendrocytes, and importantly, microglia, the primary immune cells in the brain. Extensive research has shown that as individuals age, changes at both cellular and tissue level alter many aspects of the function of these cells, including their gene expression patterns, which together lead to compromised function. This in turn results in neuroinflammation and ultimately neurodegeneration. As we know, the HIV pandemic is still ongoing, and despite all our efforts, eradication of the virus has not been possible. This stems from the ability of the virus to hide from antiretroviral therapies and immune surveillance in a latent state in many cell types, including microglia, which are the main reservoir cells for HIV in the brain. Importantly, while antiretroviral therapy is very effective in controlling viremia, it's not as effective in the central nervous system. As a result, suboptimally controlled HIV replication in microglia leads to the induction of an activated, so-called M1 state, which is characterized by the release of inflammatory cytokines. This in turn leads to neuronal damage and HIV-associated neurocognitive disorders in individuals living with HIV. Importantly, these symptoms are exacerbated as these individuals age. However, despite intense ongoing research, many aspects of pathobiology of HIV disease in the brain remain incompletely understood. My lab is focused on understanding the transcriptomic patterns and their impact on human diseases. And so as a first step towards understanding the interface between HIV-induced and aging-induced pathological effects, we attempted to characterize the aging transcriptome to get a good grasp on changes associated with aging in individuals without HIV. We used multiple model systems, including low passage number immortalized human microglial cell lines in the lab of our collaborator, Dr. Jonathan Karn, in addition to two independently performed bulk RNA-seq data sets of primary human microglia. The first one consisted of microglia isolated from the parietal cortex of healthy individuals, and the second one consisted of microglia isolated from the occipital cortex or corpus callosum of healthy individuals or individuals with multiple sclerosis. Our first step in the analysis of these data sets was to confirm that we were indeed working with microglia. We did this by looking at the expression of neuronal, astrocyte, oligodendrocyte, and microglial markers. As you can see, there is little to no expression of the neuronal, astrocyte, and oligodendrocyte markers, while the expression of microglial markers is very high, confirming that the cellular populations under study are largely microglial. To understand how the transcriptome of microglia changed with age, we performed a correlation analysis on each data set. As we all know, aging is a rather noisy event at a molecular level. However, despite that, we were able to identify over 500 genes that were reproducibly showing positive or negative correlation with age in both our data sets. Here we have a subset of the genes we identified that are mostly found in microglia. As you can see, the positively correlated genes on top are increasing with the age of the donors, while a larger number of genes shown at the bottom are downregulated with increasing age. Pathway analysis of the data sets showed that aging led to a downregulation of genes associated with a healthy microglial state. In the graph on the left, the size of the circles correlates with the level of enrichment of each pathway, and the direction of enrichment is shown at the bottom. The color of each circle indicates the statistical significance as reported by false discovery rate, or FDR. The more intense the red color, the more statistically significant the enrichment. As we can see, out of the top 4 negatively enriched gene lists, 3 of them are those capturing the gene expression pattern of normal, healthy microglia, suggesting a deep perturbation of the microglial transcriptome as a result of aging. On the left is a heat map showing some of the genes in these gene lists that are differentially expressed as a result of aging in both parietal and occipital cortex-derived microglia. And as you can see, the majority of them are downregulated, shown in blue. In these graphs, the heat map to the right indicates the change in gene expression, while the annotation matrix to the left indicates which gene lists or pathways each gene belongs to. In this case, these are genes belonging to the pathways shown in the graph to the left of the slide, as you can see at the bottom. These genes include some that are important in maintaining homeostasis in microglia, such as insulin growth factor 1 and P2RY12, both of which are negatively correlating with age. Further analysis also revealed that aging leads to an induction of core inflammatory pathways and reduced proliferation in microglia. In the heat map on the left, we see that most of the genes in inflammatory pathways are increasing with age, like the classic pro-inflammatory cytokines interleukin 1 beta and interleukin 15. We also see that interleukin 6, a proliferative gene, decreases with age. On the right is another pathway analysis graph using the MSIG-DB database, which complements the heat map to the left by showing the aging-induced positive enrichment of cytokines. Importantly, among the most negatively enriched pathways, there are multiple terms related to cell cycle and proliferative pathways such as the MIG pathway, indicating the slowdown of proliferation with age. So these results are in full agreement with published studies on aging microglia. After obtaining this overall pattern of what aging looks like in microglia, we wanted to interface this to changes in gene expression due to HIV infection. We turned to another RNA-seq database using human-induced pluripotent stem cell-derived microglia from 3 human donors. We performed a differential gene expression analysis comparing microglia infected with HIV to uninfected microglia. As we did with our first 2 studies, we wanted to verify that the data set was indeed microglia. We saw high expression of microglial markers in these cells too, while expression of the markers of non-microglial brain cells remained low. We also confirmed the robust expression of HIV transcripts in the infected cells. Our differential gene expression analysis showed that HIV infection results in strongly perturbed gene expression in microglia, similar to what we saw with aging. Around 150 genes corresponding to those expressed in healthy microglia showed greater than 2-fold differential expression after HIV infection. As a small fraction of these genes, are shown here. They're the ones showing the highest level of differential expression after HIV infection. And as you can see, the majority of these genes are downregulated. Pathway analysis on the genes differentially expressed after HIV infection also showed an increase in multiple inflammatory pathways, which was expected and has been reported before. However, interestingly, this analysis also revealed a strong decrease in proliferative and cell cycle pathways. Multiple proliferative terms, including MYC and E2F signaling, are downregulated, in addition to the key metabolic term glycolysis. So taken together, these results suggest that HIV infection induces an inflammatory state on one hand and leads to a proliferative slowdown on the other hand. This is unusual since in studied cases I'm aware of, inflammatory activation in immune cells, including microglia, leads to the induction of proliferation. This is accepting unusual cases and aging. So we thought this was an interesting effect seen after HIV infection in microglia. To further confirm this result, we checked to see how the pathways that were negatively or positively enriched in aging fare after HIV infection. Interestingly, as can be seen in this side-by-side comparison, The changes resulting from HIV infection strongly resemble those occurring in aging. We see the same cytokine pathway increasing and the same cell cycle related pathways decreasing, among other concordantly changing pathways. Finally, to make sense of these observations, we took advantage of the KEGG pathway database, which permits the analysis of core factors involved in cellular pathways. As you can see, the most negatively enriched pathway in this analysis is cell cycle, and to our surprise, we saw the cellular senescence among the pathways strongly perturbed after HIV infection. The observed enrichment has a false discovery rate of essentially 0. To take a closer look at the individual genes involved in this pathway, we superimpose the gene expression pattern on the diagram capturing the senescence pathways. As you can see from the colored key on the top right, genes upregulated in HIV infection are in red, while the genes marked in blue are downregulated. Many antiproliferative genes are activated, including GAD45 and P21, a strong antiproliferative factor. On the other hand, proliferative factors such as MYC and multiple cyclins, such as cyclins A, B, and E, were downregulated, indicating the activation of a multi-pronged antiproliferative senescence-like pathway following HIV infection. Taken together, these results are first consistent with previous results in the field that microglial aging is associated with inflammation, reduced proliferation, and perturbed gene expression patterns. They also show that while HIV infection, as expected, results in a pro-inflammatory state and perturbed microglial homeostasis, unlike other studied inflammatory states, it leads to a strong block to cell cycle and proliferation. Activation of multiple factors involved in senescence following microglial HIV infection, including antiproliferative factors, raises the intriguing possibility that HIV, unlike other pro-inflammatory conditions, may induce a functional aging-like phenotype. These results suggest that HIV infection may strongly exacerbate and accelerate the functional changes occurring with aging in people living with HIV, including functional defects stemming from loss of microglial proliferation. For my acknowledgements, I would like to thank my mentors, Dr. Saba Bulatkan and Dr. Jonathan Kahn, who've been very helpful in guiding me and supporting me with this project. And I'd also like to thank a couple members of the Karn Lab, David Alvarez Carbonell and Feng-Chun Yi, who have helped me as I've been learning more about microglia and HIV infection. Thank you.